Hello, everyone, and welcome to the first lecture on Chapter 10, Controlling Microbial Growth in the Body. Because what we have in this class are mostly pre-nursing students, I'm going to skip the section all about doing kind of lab work on decontaminating surfaces and using autoclaves and that, because that's not generally something that the registered nurses do. However, we do have to consider controlling microbial growth in the body to understand the medications that the healthcare providers will be prescribing and that we as RNs would be administering. So that's where I'm going to focus our attention. So although registered nurses are not the ones that prescribe the medications, we are the ones that administer the medications. And it is helpful for us to know the different modes of action and to understand what medications are being prescribed, why, and why you wouldn't have a medication. Well, we'll talk about it some more so you can understand like why you would prescribe this medication but not this one, or why if you prescribe 2 or 3 medications to fight an infection, you wouldn't put these 2 together, but you might put this one and this one together. Okay. Let's go over some definitions at first. What we refer to as drugs are chemicals that affect the physiology in any manner. That's just a generic term. And then here's one that is a little tricky. You've heard of chemotherapeutic agents, sometimes referred to as chemo. Usually when we talk about chemo, we're talking about a cancer treatment, but chemotherapeutic agent just means it's a drug that acts against a disease. So antibiotics can be considered a chemotherapeutic agent. And then specifically, antimicrobial agents are going to be drugs that fight off microbes, that fight off infections. Here's the absolute key to understanding how these antimicrobial drugs work. Is this concept of selective toxicity. It means that we're basically trying to kill that organism that's trying to kill us, or that is having a negative effect on us. The trouble is they're living things. We made this big fuss about we all use DNA and we use the same genetic code and all these codons mean the same amino acids and all the different species. So like that's a terrible target for a medication because it would be damaging to the host cells as well. And so what we look for are differences in the physiology between the microbe and the patient, between the microbe and the host, to target the things that are going to hurt the microbe more than they're going to hurt the host. And the more differences that exist, the more differences we can identify, the easier it is to find a target against that. So for example, with bacteria, they have cell walls. We don't. So that makes a fantastic target. And we'll go over some other targets. This is one of the trickiest ones though. Viruses. Viruses are very different than our cells. However, to do the work of copying themselves and making more of them, they use all of our own machinery. They use our ribosomes, they use our cell membrane if they need an envelope, and so it makes really hard to find a target to use against the viruses themselves. That's why it's been so hard. And so many of the drugs, the antimicrobial drugs, are antibiotics. They're medications that are used against bacteria because bacteria are so different from us. And viruses, I think of viruses as using like human shields. If you've ever seen those TV shows where there's a hostage situation and, the police, there's a SWAT team and then there's the kidnapper or whatever. And he's hiding behind the kid. That's what viruses are. They're doing all their bad stuff, but they're hiding behind our ribosomes. They're hiding behind our cell membranes. And so it just makes it a lot harder to fight against viruses. It doesn't mean that it's not worth trying, but there aren't as many medications available yet. A little bit of history about the antimicrobial agents. Paul Ehrlich is the guy who kind of gets the credit for articulating this idea of finding a medication that can target a living thing that's trying to live inside of us without killing us. And he referred to them as magic bullets. They were things that would be able to, knock out the infectious agent without knocking out the host. His one of his contributions was using arsenic. You've heard of arsenic, that's poison, but it's more poisonous to syphilis than it is to us. However, it has to be finely titrated because it can be harmful to the patients as well. And so there's a fine line to walk with arsenic dosing. But at the time, that was the best people had. Nowadays, people get a syphilis infection, it's an antibiotic, no big deal. At the time, people would die of syphilis. They would have dementia related to syphilis. I mean, it was really bad news. And so arsenic was an improvement on that. But yeah, so Ehrlich gets the credit for this idea of magic bullets. And then Alexander Fleming, he was the dude who is credited with discovering the first antibiotic or the first antibacterial medication. And it's genius. So penicillin is the compound, and it's called that because it was released from a fungus called Penicillium. That's the genus name. And if you can think about this ecologically, fungi and bacteria are often competing for resources. Like just think, rotting fruit. And so if a fungal spore arrives or bacterium arrives and they both start growing, they can try to compete with each other, right? And so it was an observation that Fleming made of an area where bacteria weren't growing, where there was this fungus growing. So that was in the late 1920s, but penicillin didn't get widely used until the 1940s. It wasn't actually until World War II. And it's one of the reasons that the Allies were successful was because during the war, more soldiers could be saved from infection than the other side could save from infection. Okay. Let's see, Gerhard Domach, he discovered sulfonamide. This was the first antibiotic that was used more widely in the 1930s. So although it was discovered after penicillin, penicillin didn't really get distributed widely until the 40s. So there's these so-called sulfa drugs came in a little bit earlier. And we'll talk a little bit about the mechanisms of action of these sulfa drugs and how they're different from penicillin. What I want you to see though, or hear is, hey, if you look into nature, these microbes have been battling each other for millennia. So looking to them to see, hey, how do you fight bacteria? What compounds do you have that we can tap into? Very, very clever. And then finally, Waxman coined the term antibiotics. The idea is like against living things, against things that are going to cause disease in us. Nowadays, this term antibiotics typically comes to mean antibacterial drugs, but generically it means against living things. But this was again, this idea of like going to nature, finding a species that has to fight off bacteria and see how they have evolved a mechanism to fight off bacteria. So Streptomyces is a fungus, and it can produce a compound that fights off bacteria. Let's see, I want to move a second, trying to move something. And is that the last thing on my screen? Yeah, okay. And then finally, here's an image of what we're talking about. So this is a petri dish, Staphylococcus aureus. That's a bacterium. It lives all over our skin, and it's very common in the area. And it's been streaked onto an agar plate. And here's some fungus growing. And check out this zone of inhibition. Like clearly, the bacteria got streaked onto this plate, and yet we do not see growth in this halo around the fungus. That, my friends, is how this was discovered. It wasn't on a petri dish, but it was like people just being observant about their environment and going, huh, where the fungus grows, the bacterium doesn't grow. How come? And that's what led to the discovery of the drug penicillin. At the time, there was no idea of what the mechanism was. You just made the observation empirically, like, look, stuff doesn't grow here. All right, some more definitions. Semi-synthetics, that's the most common kind of functional antibiotic that we use nowadays. So it's something that was discovered from nature. That is, okay, what is it that the penicillium fungus is making, and then alter it a little bit chemically so that it's more stable. So like you put it in a pill and put it in a bottle and leave it on the shelf, for months on end. And something that's easier to administer than the naturally occurring ones. Like we don't have to grow a fungus in our patient to fight off the bacterium. We're just isolating this compound and then adjusting it a little bit to make it more shelf stable, if you will. So that's semi-synthetic because it's partly engineered and partly based on a natural organism, a natural compound made by an organism. And then there's fully synthetic antimicrobials. These are not as common, but they're completely synthesized in the lab. And how you would do this is you figure out what's the structure of the bacterium, and then what protein structure would we have to build that would, block that one. These are more costly. The semi-synthetics just give it's we're copying, we're plagiarizing the other organisms in the universe that have fought off bacteria before and seen how they are doing it, and we copy that. All right, so there's a number of different modes of antimicrobial action, and it all boils back down to what are the structures or what's the physiology of this microbe that's different from the host. And that is like how we can target a medication. So for example, the cell wall of this microbe, we don't have cell walls. That's a perfect target. The plasma membrane, that one's trickier because we have plasma membranes, but there are some subtle differences that we can target. There could be a metabolic pathway, that is a series of chemical reactions that happens in the microbe that we don't have that series of chemical reactions, and so we can target that. Inhibition of protein synthesis, that's a tricky one because we synthesize our own proteins, right, using our ribosomes. But remember, bacteria have ribosomes that are a little bit different from ours, so that might be a good target. And then finally, inhibition of DNA and RNA synthesis. And once again, we use the same DNA, same RNA. So what we have to target is something like the enzymes involved are maybe slightly different from each other. And then those can be a target. And why I'm going through this in all this detail is to remind you, this, my friends, is why we study physiology. This is why we need to know all of these details, because the more we understand about the differences, the easier it is to develop drugs to combat these things that won't have a ton of side effects for the host, the ton of side effects for the patient who has to take these medications. All right, so as I've mentioned, this idea of selective toxicity is a drug that's going to be more harmful to the microbe than it is to the host, and that the vast majority of the drugs, chemotherapy agents against microbes, they're actually just antibacterial. And again, the reason is because bacteria are so different from the eukaryotic cells that there's lots of different targets to go after. There aren't as many drugs to treat eukaryotic infections. So to treat fungal infections or to treat worm infections, there are some effective treatments, but there aren't as many targets. And much less, holy smokes, antiviral drugs. There's been progress, don't get me wrong, there are definitely some antiviral medications, but not nearly the number of antiviral medications as there are antibacterial medications. And again, it's not that viruses can't hurt us, it's that they're using us as their human shields, and it's harder to fight off a viral infection with a drug. Here is a cartoon representing all of these different things. So inhibition of cell wall synthesis, we said that's going to be a fantastic target. And so look at this list of medications. I mean, there's so many that target this structure because we don't have that structure. Cell wall is not a thing that our human cells have. And so some of the drugs like this, penicillin, now we know that those fungi that make penicillin, that compound impacts the bacterial cell's ability to make a functional cell wall. Now there's lots and lots of different ones. The ones I want you to know, so penicillin, cephalosporins are also this, vancomycin, that's a big one. Isoniazid, ethambutol, are all targeting the cell wall. Now, these are all for bacterial cell walls. There is an echinocandin that targets the cell wall of fungi, but the rest of these are all antibacterial medications. Then a second fantastic target is inhibition of protein synthesis. And we've said, wait, protein synthesis is something that we have to do. It's going to be a little bit tricky. So often the target of these is actually the ribosomes that are doing the translation step and making the proteins. So some examples of medications like that are aminoglycosides, tetracycline. I think you've heard of tetracycline. It's a pretty common medication. Chloramphenicol, macrolides, you might not recognize that term. There's erythromycin is a medication that you might have heard of. It's an example of a macrolide. Let's see, what else? Disruption of the cytoplasmic membrane. A couple of different ones. I didn't include polymyxins. I'm not quite sure why. Oh, I know why. So polymyxins, they act on the plasma membrane, and I'll try to call it a plasma membrane, but cytoplasmic membrane, cell membrane, it all means the same thing. They're different from cell walls. That's peptidoglycan. The cytoplasmic membrane or plasma membrane or cell membrane, all of it referring to this phospholipid bilayer. So polymyxins can act on the phospholipid bilayer of bacteria. The polyenes are an antifungal. They can act on the phospholipid bilayer of fungi. And let's see, one more. Inhibition of a general metabolic pathway. That is something other than the things that we just talked about. The one example that I really want you to know is sulfonamides, and we'll get to it and I'll go over some detail, but basically it inhibits the pathway that the microbes have for making folic acid or folate. You might know it as vitamin B9. It's absolutely required for making DNA in some of the microbes. We humans also need folic acid, but we don't make it ourselves. We actually get it into our diets. That's how we get it. And so the sulfonamide doesn't impact us. Okay. And there may be some that inhibit DNA or RNA synthesis. I am going to mention a couple of these later in the lecture. And inhibition, attachment to recognition of the host. But the ones with yellow boxes around them, those are the ones that you're going to hear about the most, and you as an RN would be administering these drugs. Okay, so to review, I will finish with this. This is the mechanisms of antimicrobial drugs. There's going to be inhibition of the cell walls. We're going to look at inhibition of bacterial cell walls, or lots of different kinds of medications, and inhibition of fungal cell walls. Then the next target is a plasma membrane, cytoplasmic membrane, cell membrane, means the same thing. We'll look at disruption of the bacterial plasma membrane, and the one example was the polymyxin. And then we'll talk about the fungal plasma membrane. And there are a couple of different kinds of drugs that can target that. Then we'll move on to inhibition of protein synthesis and inhibition of metabolic pathways. And both of these, we're talking primarily about medications that impact bacteria. And again, why don't we use these same tricks against viruses? Because viruses are using all of our machinery to do their protein synthesis. And so there's no target that we can use against the virus that's not going to impact the human cells. That's the big problem there. And the same with the metabolic pathways. Viruses don't have separate metabolic pathways that are independent of our metabolic pathways. And so it's not a great target for that. All right, so that is the end of part one. I'll see you over in part two.